Hello students. So I'm going to discuss one of the questions, that is, this is a segment of DNA CGAT GGCTA. Now the question is asked that what is the messenger RNA strand for this DNA segment? And the options given to it are: option A is GCUA, CCGAU, option B is UCT, ACCGUA, and option C is CGAT, GGCTA, and option D is ATC, GGT, AGC. So these are the four options. And the segment of DNA has the sequence, here I am highlighting with red color. The sequence is CGAT, GGCTA. So what can be the transcript of messenger RNA? So coming to the explanation first of all. So let's move to the explanation. Now first I would like to briefly describe that for a DNA segment, DNA is a double-stranded segment. It means and the strands are held together by the hydrogen bonds between the nitrogen bases. Okay, and as we know that adenine base always pairs with thymine and guanine always pairs with cytosine. Guanine pairs with cytosine with 3 hydrogen bonding, and adenine pairs with thymine with 2 hydrogen bonding. So this is about the complementary strands of DNA. Now when we talk about the difference between the DNA and RNA transcripts, so DNA and RNA varies in the nitrogen base. And what is the base that makes a difference? That is the presence of uracil. Despite of containing thymine that is present in DNA strand, RNA always contains uracil. So it is clear. So whenever the strand is complementary, whenever we have to copy something from DNA strand, then RNA always pairs with uracil despite of pairing with thymine. So suppose this is a DNA transcript, and this DNA transcript contains the sequence ATTCT sequence, then RNA will contain UAAGA sequence. So it is clear. So despite of containing thymine, if we have to take out the complementary DNA, then what will be the result? Complementary DNA will be thymine, then this thymine will pair with adenine, this thymine will pair with adenine, this cytosine will pair with guanine, and this thymine will again pair with adenine. But at the same time, when we talk about messenger RNA, then this code changes. Adenine will pair with uracil, thymine will pair with adenine, thymine will pair with adenine, cytosine will pair with guanine, and thymine will pair with adenine. So this is the application that we have to apply in the given question. Now moving to the question. So now here you can see the segment of DNA is C. So now I am taking another color. Suppose I have taken green color. Now C will pair with which code? G nucleotide base. G will pair with C nucleotide base. A will pair with uracil because it is talking about messenger RNA here. Thymine will pair with adenine. Guanine will pair with cytosine. Guanine again will pair with cytosine. Cytosine will pair with guanine. Thymine will pair with adenine, and adenine will pair with thymine. So the overall sequence that we got here, adenine will pair with uracil. So the overall sequence we got here is GCUACCGAU. Now it is asked that what is the messenger RNA strand for this DNA segment. So when we look at the option A is exactly the same option that we have found, that is GCUA-CCGAU. When we take the option B, when we see the option B, then there is no exact complementarity between the sequences. So option B is entirely invalid. This is exactly invalid option. When we take the option C, so yes, C cannot pair with C in its complementary messenger RNA sequence, so it is also false. So first, when we look at the first nucleotide itself, it is wrong. And when we take the example of D option, then here you can see C can not pair with A, so it is also false. So only one option is correct, that is the option A. So now we are taking another color to select the answer. This color is red. I am highlighting the exact answer with red color. So the overall answer to this question is option A is the correct option.